New law. Iowa to expand DNA collection from criminals. Gene behind aggressive lymphatic cancer identified. Genetics is always in the headlines. Every day there's a new story or journal article involving some of this jargon. But what is this stuff anyway? Why is DNA so important? What do all of those words mean? To understand all this, let's begin with the basics. DNA stands for deoxyribonucleic acid, which really just describes it as a chemical. DNA has a sort of backbone made out of a chain of sugar and phosphate units bonded together. The sugar is called deoxyribose, which is where the deoxyribo part comes from. DNA is found in the nucleus of the cell, which is where it is stored and protected from damage. That's where nucleic comes from. DNA is an acid because the phosphate groups release hydrogen ions in solution, lowering the pH slightly. The sugar-phosphate backbone is bonded to molecules called nitrogenous bases. There are 4 kinds of bases in DNA: adenine, guanine, thymine, and cytosine. Each base is bonded at one end to the sugar-phosphate backbone, and on the other end to another base. Adenine likes to bond with thymine, and guanine likes to bond with cytosine. But what function does DNA serve? The function of DNA begins when the 2 strands are separated. Now, an enzyme called RNA polymerase finds a particular spot on one of the strands and starts copying it. It uses RNA, which is just like DNA, except that it uses a sugar called ribose instead of deoxyribose in the backbone. RNA is slightly different, but shares basically the same structure. RNA polymerase adds complementary bases to match the template strand, so that the RNA strand that is formed is the same as the coding strand. The major difference is that RNA has a base called uracil instead of thymine. Uracil and thymine are almost identical, so we can think of them as being the same nitrogenous base. After this RNA strand has been produced, the DNA strands can be joined together again, and the RNA leaves the nucleus to find a ribosome. The ribosome reads the nitrogenous bases in clumps of 3, called codons, and each codon codes for one of 22 different amino acids. It keeps reading all of the codons and joining the amino acids together so that in the end there is a long chain of amino acid. These chains have to fold in a particular orientation, and then we call them a protein. Proteins are pretty much responsible for everything that happens in your body. Virtually all of the chemical reactions that keep you alive depend on proteins, including, for example, the way your body uses oxygen. In fact, even the reactions that transcribe DNA into RNA are facilitated by proteins. Now we can define the word gene. A gene is the segment of DNA that codes for the amino acid sequence of a particular protein. An allele is just a particular version of a gene. For example, there is an allele for brown eyes and an allele for green eyes. Different alleles of the same gene produce similar proteins that may vary slightly in their function. Back to the story. When a cell divides, all of its genetic information must be copied and passed on to both daughter cells. This is very similar to reading the DNA. The strands are separated, and a new DNA strand is made to complement the old strand. This is basically the same process as before, except that the entire length of DNA is replicated on both strands. The cell must copy every single gene so that the daughter cells can both be functional. At this point, you probably have some questions. Do the mechanisms inside the cell always copy the DNA perfectly? What happens if there's a mistake? What kind of mistakes can it make, and how does this affect the daughter cells? To find out, check out the video on mutation. Thanks for watching. If you have comments, suggestions, ideas, or questions, feel free to transcribe them to the comments below. I would hate to replicate what other videos do, but could you do me a little favor and subscribe?